Hi everyone. Today we are going to continue on our journey of learning about different genetic engineering technology, and we're going to focus on genomics itself and recombinant DNA. So just to make sure we're all on the same page, I'm just going to give you our working definitions of these two key terms. So a genome is the entire genetic makeup of an organism. So this is every single nucleotide sequenced so we can understand exactly what our genetic makeup is. And then a gene is a specific DNA sequence that encodes for a specific protein. Now, I mentioned when we worked on one of our previous assignments that I was going to give you just kind of like an overview of the Human Genome Project, just to make sure that in your notes you had exactly what I wanted you to have so that you would be totally prepared for the test. The test is not for a while, so nothing to worry about, but I just wanted to give you this and kind of go over the key points. So this is just a general timeline. We've talked about some of this before, right? So we've got our first gene map created, and then Francis Crick and James Watson did their whole DNA thing where they discovered the molecular structure of DNA. Then we got Sanger coming in with sequencing DNA, and then we're talking in the 80s. So in the early 80s, we sort of automated DNA sequencing, and the idea of automating this sequencing made it quicker, right? You could do more in less time. And then it started to bring like this idea to biologists that were like, well, okay, I like where this is going. Let's sequence humans. And that's sort of where this whole idea started. And then in 87, an early project was started by the Department of Energy. By the following year, Congress decided to fund this project because they liked this idea. So NIH and DOE decided to coordinate efforts, and in 1990, an initial plan was set. The plan was to take this to take the next 15 years to sequence the entire human genome. However, they were able to finish earlier, and in 2001, they announced that the genome was 90% sequenced. This entire project led to new technologies and advances in gene sequencing, and that's actually why they were able to finish this earlier, because everybody working on this same project for over a decade led to ways to make this process easier. And then in April 2003, the human genome was completely sequenced. And now we use this still, but this was like the first time we were able to actually understand the entire genetic blueprint of a human being. This is incredible. So you may be asking yourself, okay, that's cool, but where'd they get all this DNA to do all this sequencing? And actually, it was just volunteers. People were like, yeah, that's cool, I'm into it. And they just provided DNA samples. Tons of them, right? Several volunteers. It wasn't just one person because they needed to make sure that they wanted to determine which sequences were the same in every human and which ones were unique in different humans to determine where the actual genes were. And then, of course, you will never know who actually volunteered for this because it was a very secure process where volunteers were not revealed, and all of their DNA that they were donating to this project was collected and maintained in a very specific way, so no one could find out who the volunteers were. And then just some like really cool outcomes. There's about 4 million DNA variants per individual, which means that out of the billions of base pairs that humans have, only about 4 million of them code for the differences that you see in humans itself. So that's not a lot. That's a very, very small percentage of differences between human beings, considering the differences that you physically see between human beings. A couple other cool things is after they sequenced this, they realized that about 2% of people in the world are likely to succumb to Alzheimer's disease based off of the genes that they have. About 1% of us have two copies of a small deletion in a gene, which makes you pretty much immune to HIV. And about 7% of the world don't have a functional CYP2D6 enzyme, which basically allows, well, it doesn't allow, it makes it so that codeine has no pain relief. So this is talking about if you were going in for surgery and you were going to have codeine to limit the pain, about 7% of the world, that would have no effect on them. So we've sequenced the human genome. So now what? Well, now that we've sequenced this, we can apply this knowledge pretty much everywhere. So our next steps as scientists is to understand how the genome works. We know what it looks like, but we don't actually know what each gene is creating. So identifying the genes, how they work, and the differences between individuals and different species. That's kind of like the next big piece that we're trying to work on. Of course, we're trying to understand how diseases work and the genes behind the disease, and then any form of advancing medicine or improving the effectiveness of medicine, especially individualized medicine. That would be like a goal that is currently, it's a goal that's set, but no way you're going to be able to do this within the next couple of years. So this is like more of like a future goal. And then again, applications of this DNA technology. So there's medical applications where you can locate the genes in disorders, diagnose and treat genetic disorders, or use gene therapy. There's pharmaceutical applications where you can create new drugs and produce large amounts of medically important proteins like insulin. And then you can use this for forensic and genetic profiles, use it for environmental cleanup, and for agriculture. So one of the big ways that we use this information is in the form of what's called recombinant DNA technology. So recombinant DNA, this is like our big focus point of this lecture. So recombinant DNA is combining DNA from different sources into a singular DNA molecule. And we do this with the use of restriction enzymes. So we're going to get a little bit more into restriction enzymes, but just to make sure you understand, this would be like taking DNA from one animal, whatever it may be, a salmon, I don't know, and inserting it into a bacterial DNA to form one new type of DNA, and then inserting that new DNA back into the bacterial cell or a new bacterial cell, it doesn't matter, and then letting that bacterial cell replicate. So then you would have tons of copies of this new type of DNA. So the way that restriction enzymes work is that you are cutting DNA. So, right, you have some sort of recognition sequence. Typically, it's in the form of a palindrome, which means that it is the same letters forwards and backwards. So you can see that here, it's AATT, and then backwards is AATT. So it's the same thing forward and backwards. And so this restriction enzymes cuts this DNA at a specific sequence, typically called a restriction site or a recognition sequence. Once it breaks open, you're left with these kind of prolonged, protruding ends. They are called sticky ends. And they're called sticky ends because DNA will stick to them. So after the restriction enzyme cuts the DNA into fragments, these sticky ends look for complementary DNA. So the DNA from another source is added that has those same complementary base pairs. They sync back up, they stick together, and then DNA ligase comes in and actually like paste those fragments together to make one continuous piece of recombinant DNA. Now, these enzymes are found in bacteria, and there's a lot of them. And so the enzymes are just named after organisms that they're found in, so they have like totally wonky names. And I don't need you to know that. I don't need you to memorize any of the names. I just want you to realize that you might see them and you'll be like, what? How do you say that? And it's like, it doesn't matter. They're just named after different bacteria. And so, like I was just saying, restriction enzymes come from bacteria. And originally, the actual purpose of restriction enzymes is to kill viruses. It's to prevent viruses from being able to take over a bacteria with their viral DNA. And so the enzymes in the bacteria attack the viral DNA and break it up into useless fragments. Well, scientists saw that bacteria could do this naturally, and they were like, I'm gonna take that, I'm gonna use it for myself. And lo and behold, they did that, and that's how we got recombinant DNA and now use it for a variety of different things. One way that we use it is in transgenic organisms. I'm sure that you were probably sensing this from where we were going with our assignments so far, that we were eventually going to get into genetically modified organisms, or GMOs. And the way that GMOs are formed is using restriction enzymes and recombinant DNA. So what they do here is that scientists add the DNA, whatever it is that we're interested in using, whether it's the human insulin gene, whether it's some sort of pesticide, it could be anything. But whatever DNA the scientists are interested in, they plug that into a bacterial plasmid. And a plasmid is just a circular piece of extra DNA that's found in bacteria. And so looking at this picture over here, you can see that the human DNA, we have our DNA of focus, right? Our interesting DNA that we want to use in this bacterial plasmid. So we break that up using the restriction enzymes, and we insert it into our bacterial plasmid, which is this purple circle. Once it's in there, DNA ligase comes in and completes everything, right? It makes it one smooth plasmid again, right? We've made it a smooth DNA plasmid, and then it gets inserted back into a bacterial cell. And the bacterial cell doesn't know any better, so it just replicates, does what it does best, and just replicates and replicates and replicates, and then you're left with millions of copies of this new recombinant DNA, and you can use this recombinant DNA in a variety of ways. And we'll get into that, but this is the process that scientists have used for years and are continuing to use to make transgenic organisms, or kind of more commonly known as, in like the public sector, as GMOs. So the first commercial healthcare product to use this technology is human insulin. So the insulin gene was targeted and separated and inserted into a plasmid. After that, that recombinant DNA was put back into the bacterial chromosome, and again, the bacteria were just left to reproduce, and now we can extract pure insulin, pure human insulin from this DNA, or from these bacteria, because of this DNA, and now we can use this pure insulin for anything that we need insulin for, but a lot of it is used for people who have diabetes. On the flip side, like creating recombinant DNA and finding all of those cool things, like making all of that human insulin so that we can use it for diseases, that's a great use of recombinant DNA making transgenic organisms. Awesome. On the flip side though, that's not the only thing we use it for. So genetically modified organisms are everywhere. It's in bacteria, it's in plants, it's in animals, it's everywhere. Sometimes it's okay, like on this side we're talking about types of corn and genetically modified corn, and what this specific corn was about was to make it more hardy and to make it more resistant towards diseases. Okay, cool. That'll help with, people going hungry. I get that. But then we have, sometimes they just, or not sometimes, they did this multiple times. They added this green fluorescent protein And they just added it to random organisms just to see what would happen. And so you can kind of see that, like, okay, well, actually, that's kind of cool looking in the jellyfish. And, like, yeah, I'll give you that. It's kind of cool looking, but why? Why are we doing this? What is the purpose? Why did we add it to this pig so that that pig can glow green? Why did we add it to these mice? We're doing things because we can, but are we doing things because we should? And that's a question that I just want you to think about. And that's kind of the driving question behind are genetically modified organisms okay? Are we doing things because it's a good idea, or are we doing things just because we can and it's fun? Some ways that we've been able to apply genetic engineering in a positive way are listed here. So we can improve crop plants with nutritional qualities, right? We can increase the amount of nutrition, which means that you can eat less but gain the nutrients that you may need faster. We can increase animal growth. Again, this helps with hunger around the world. If fish are bigger, they feed more people. We can also engineer microbes to help degrade toxic waste. There are microbes around that eat plastic. They've actually evolved to eat plastic, and so we can take that and expand on it and then start to remove some of the excess plastic in the world. So that could be cool. But the production of these useful proteins, so making these useful changes into these organisms, is the primary driving force for the use of recombinant DNA. So all of these studies, all of these experiments, everything that the scientists are doing is just to produce useful proteins to help humankind. That is their goal. And then the last thing I want to talk about today is reverse genetics. So what we've been talking about kind of so far this year is that you can change a genome through mutations. So mutations are traditionally provide the raw material needed for analysis. So you can see that you have a wild type of something, and then a mutation will give you a mutant phenotype. And then from there, you can kind of recognize the mutant gene, then identify the wild type allele and its normal function, right? You typically start with the mutant phenotype to recognize that there even is a mutant gene, and then identify the wild type allele and its normal function. But recombinant DNA has made possible a different approach. So you clone wild type genes, right? You start with the wild type, and then you intentionally mutate them in different ways. So you're taking your normal genes that would be found in a wild type organism, and you're purposely messing with them. Once you purposely mess with them in whatever way that you want to, you introduce that back into the organism, and then you study what happens. So it's sort of like the reverse, go figure, they call it reverse genetics, but it's basically the opposite of how we used to do it. So now instead of waiting for mutations to occur, we're just forcing mutations to occur and then studying what happens. This last image is just a sort of overview of everything that we talked about, right? We've got our bacteria, we've got our DNA of interest, we put the gene of interest into the plasmid, put it back into the bacteria, let that replicate a bunch of times, and then down here is sort of our various research, like applications, how we're using this research. We've got pest resistance, we've got cleaning up toxic waste, which is sort of what I was talking about earlier with the plastic, but it can also be used for like oil spills and stuff, dissolving blood clots in the heart, and then human growth hormone can help with stunted growth of humans, of course. So there are a lot of really awesome applications to this research. The question I want to leave you with is, should we be doing this? Are we playing God? And that's a discussion we'll have in class. All right, I'll see you next time.